Hello, everyone. Good afternoon. Nice to see you all. I am very upset that I missed this morning session today. I'm very thankful to SRF for accommodating my travel snafu, we'll say. And the goal of my talk today is to update this audience about the different preclinical therapeutic approaches that we're pursuing at our center at Penn Medicine and CHOP, as well as some of the resources that we've developed that I hope can serve this community. So the brief background, for those who don't know me, all of my background, training, first 5 years of the lab was on molecular cardiology. Now the majority of the lab actually works on the brain, and I'm helping to lead a center on neurological disorders. And that, of course, was inspired by the birth of my amazing and beautiful daughter, Lucy, who is shown here and who was quickly diagnosed with STX-BP1 related disorder, which shares a lot of common features with SYNGAP. So this is Lucy. This is our family. In the top right there, Lucy is enamored with her new baby brother, who's about 2 weeks old. And so I really need to thank my heroic wife, Erin, in the picture there for letting me come to this. And I'm so excited about the night's sleep tonight in the hotel. And Erin is a little bit jealous. I'm not going to go through Lucy's story, but suffice to say, she's made it very clear to me how I can do the most good in this world. And my family really aims to have a transformative impact in this space. And so fast forward, and this is the center that formed last year. We have a translational research arm, which is led by myself and Dr. Bev Davidson, and a clinical research arm led by Dr. Helbig that you all heard about a bit earlier today. And our goal is to both develop and to trial new gene-targeted therapeutics for genetic neurological disorders. And right now, we're exclusively focused on STX-BP1 and SYNGAP1 disorders. Okay, so how can we boost SYNGAP levels? Well, we have about 5 or 6 different strategies in pursuit that target SYNGAP at the DNA, the RNA, at the protein level. I'm not going to speak at a length about 2 of these programs today because they're with industry partnerships, not yet ready for prime time. But I will talk about a variety of others. And so I'm going to start at the DNA end of the spectrum. And I wanted to update on a promising CRISPR activation approach to boost the transcription of SYNGAP1 that's being led by Liz Heller at Penn and Laura in her lab. So the Heller lab has tested their strategy on this SYNGAP1 haploinsufficiency mouse model that was recently characterized by the Hugeneer group. And the Heller lab has confirmed in their hands that this mouse is indeed haploinsufficient for SYNGAP. It exhibits hyperactivity, and it has elevated ERK signaling in line with past reports. Now, to try and upregulate SYNGAP in this context, they've generated guide RNAs, those little red hashes at the bottom there, that target 2 different promoter regions on SYNGAP based on epigenetic profiling. And those guide RNAs help direct a dead Cas9 construct that is fused to a potent transcriptional activator. And they use a powerful experimental design doing dual injection of lentiviruses, one that encodes the CRISPR-A machinery, one that encodes the guide RNA that targets SYNGAP into one side of the brain, and in the other side of the brain, they inject a non-targeted guide RNA with the CRISPR machinery. So you have this powerful paired approach within the same brain. And promisingly, with this approach, they see pretty nice upregulation of SYNGAP1 mRNA and protein on the side of the brain that received the SYNGAP guide RNA relative to the control, and with preliminarily a more robust protein upregulation in the haploinsufficient mice compared to wild-type controls. Now, encouragingly, this was also associated with a reduction in ERK signaling, suggesting potentially some functional benefit of this SYNGAP restoration. So the group is currently evaluating other behavioral metrics in these mice, as well as extending these results to targeting the human gene sequence, which I'll come back to at the end. Okay, so now moving down the central dogma to the pre-mRNA, mRNA level, I'm going to briefly update on this splice-switching antisense oligonucleotide strategy to increase SYNGAP expression. And I'll do this rather quickly as this is published work from Janine and Alex within my lab. So we and others noted several years ago that there is an alternative splicing event on SYNGAP1 exon 11. This was actually first published from the folks at Stoke Therapeutics, I believe. And this alternative splicing, it leads to a certain number of SYNGAP1 transcripts that would include a premature stop codon that triggers nonsense-mediated decay, or NMD. And so in thinking about what drives this alternative splicing and how you could block it to upregulate SYNGAP, we observed that there were several binding sites for this protein PTBP, which is an RNA splicing factor in the brain, just upstream of this alternative splicing event. So we can test whether PTBP is controlling that alternative splicing by knocking it down in human iPSC neurons and assessing SYNGAP splicing. And so if we knock down PTBP in red, we see we can indeed reduce this non-productive alternative splicing that will trigger NMD while getting more of the productively spliced SYNGAP1. And importantly, corresponding to that improvement in splicing, we get more SYNGAP mRNA and protein when we knock down PTBP. Now, we don't want to broadly disrupt PTBP due to the collateral damage it would cause. We want to just disrupt its interaction with SYNGAP1. So to this end, we mapped where PTBP is binding across the transcriptome in human brain samples. And indeed, if we zoom in on just this region on SYNGAP1, we see there are these nice peaks that indicate PTBP binding right near this alternative splicing event. And we see these two key sites of PTBP regulation, which we'll call site 1 and site 2. So now our job is to see if an ASO, a steric blocking oligo, can kick off PTBP from binding there to improve SYNGAP splicing. So we walked ASOs all around that region, and we found lead ASOs that can hit both of those sites. And you see that with increasing concentrations of either these site 1 or site 2 targeting oligos in green and blue, respectively, we can increase the productive splicing of SYNGAP1, increase its mRNA levels in HEK cells. And actually, you get this huge increase with this site 2 oligo, about a 10-fold elevation in SYNGAP mRNA. Now, finally, these ASOs are increasing SYNGAP protein levels also in this dose-dependent fashion. So we've tested these ASOs in multiple patient-derived iPSC neuron lines. And this is actually the first resource that I wanted to make the group aware of. So we have patient-derived lines, but we have also generated what we call an allelic series of iPSCs, where we've engineered in different truncating variants and missense variants, which you see schematized here, into a common genetic background. And this can reduce background variability in your iPSC neurons and will hopefully allow us to assess functional phenotypes that can be different between different mutation types. So these lines are undergoing electrophysiological characterization now, but if they could benefit your research program, please don't hesitate to reach out. Okay, but back to this splice switching story. If we treat these SYNGAP patient-derived neurons with site 1 or site 2 targeting ASO, we can restore the SYNGAP1 mRNA levels. But you'll note here, this is about a 2-fold upregulation in neurons with our best performing ASO, where the same ASO led to about a 10-fold increase in the HEK cells. And we think that is because this is a developmentally regulated splicing mechanism. So you have high levels of PTBP, high levels of non-productive splicing in a non-neuronal or an immature cell, which is probably restricting SYNGAP expression where it's not needed. But as the neuron matures, PTP levels drop, you increase the splicing and expression of SYNGAP as needed in the mature neuron. But this has an important therapeutic implication because with a limited amount of alternative splicing in the mature brain or in a developed brain, we expect there may be a low ceiling for SYNGAP upregulation with this approach. In our hands, maybe a 1.5-fold change in protein expression in the mature brain. And this matches what was published, around the same time from the Zhang lab at Chicago, where they used a genetic approach to delete this alternative splicing event. So the key question I wanted to pose to the crowd with this slide is, what is our best data that informs on the minimum effective dose of SYNGAP that could confer meaningful benefit? Is 1.5-fold enough, and is a partial rescue really worth pursuing? Okay, so moving along to the last approach that I wanted to highlight, and this is a translational read-through strategy that's being pursued with John Lewick's group at the University of Rochester, and this data was generated by Alex in my lab. So John Lewick actually helped pioneer the use of what are called anticodon engineered transfer RNAs, or ACE tRNAs, to allow targeted read-through of premature termination codons, which is the genetic underpinning for around 10 to 20% of genetic epilepsies. So in brief, if you have a mutation where, for example, a glycine residue is mutated to create a premature stop and thus lead to nonsense-mediated decay or a truncated protein, you can deliver these ACE tRNAs, which will recognize the mutated codon, but deliver the canonical and appropriate amino acid to allow reading through of that premature stop and restoring expression and preventing NMD. So we've tested these in a premature stop SYNGAP patient line, this R1240X mutation, which indeed demonstrates about 50% expression of SYNGAP1 mRNA and protein, indicating that the mutant transcript is being degraded by NMD. And so what's cool is that if we deliver this ACE tRNA encoding the appropriate arginine anticodon, it indeed restores SYNGAP1 mRNA in a dose-dependent fashion. And we think this is specific to reading through that mutation, because if we deliver the exact same construct to a corrected line that lacks the mutation, you see no effect on SYNGAP levels. So we've now moved those constructs to an evolved AAV capsid that's being developed by the Davidson lab, which we're pursuing to improve the delivery of genetic material to the human brain. And promisingly, we see again this dose-dependent increase in SYNGAP1 mRNA and protein levels with this in this premature stop line with this potential clinical candidate. So to move these studies forward, there's a mouse model under construction of a SYNGAP1 harboring a nonsense mutation. Okay, and so the last point I wanted to make is on this topic of mouse models. So most of the strategies we're working on target the human SYNGAP gene. So to allow in vivo testing, we've also built a SYNGAP1 humanized mouse model. And we've made this freely available to anyone in academia or industry. So here we replaced the entire endogenous mouse SYNGAP1 locus with the human gene sequence. So this is around 100 kb replacement. This includes upstream regulatory regions, intronic regions, and downstream regions. And amazingly, this mouse actually survived. It worked out well. They breed and survive normally. They have no mouse SYNGAP mRNA and only human SYNGAP mRNA, and they make human SYNGAP protein in the mouse context just fine. And we've crossed this humanized mouse with the existing SYNGAP1 heterozygous mouse model to make a humanized haploinsufficiency model, which I think is really the ideal model to test functional rescue of clinical candidates in vivo. These mice are indeed haploinsufficient, and while the phospho-ERK is a bit noisy in our hands, it does appear to be similarly elevated in this mouse model compared to the classical heterozygous mouse model. Now, Brandon Brown in the Davidson lab, who's actually sitting in this audience, has also done some really nice behavioral phenotyping of these mice. And they indeed recapitulate the hyperactivity that has been seen in other haploinsufficiency models. They exhibit an increase in stereotype behaviors, and they have this very nice learning and memory deficit, quantified on the right there using the Barnes maze assay. That phenotype I really like. It looks quite robust, and we hope this will give us good sensitivity to be able to detect cognitive improvements with therapies that are targeting the human gene sequence. So the EEG phenotypes are scheduled to be assessed in these mice, and we will update our preprint on bioRxiv with this data as soon as it is available. So with that, I will conclude by saying our center is developing DNA, RNA, and protein-targeted therapies aimed at boosting the expression of SYNGAP1. We've developed an allelic series of iPSCs, as well as a humanized haploinsufficiency mouse model that we think should facilitate preclinical testing and that we would like everyone in this room to take advantage of in their research programs. We also show proof of concept for CRISPR activation, translational read-through, and multiple ASO approaches to restore SYNGAP expression in the context of haploinsufficiency. But to me, establishing a minimum effective dose for functional rescue of translationally relevant behaviors is really the critical next step for us on the translational side. With that, I will close by thanking the amazing team that I am so fortunate to work with, and thank you guys for your attention and time.